Ah, du coup, c'était latino.
Ah ouais.
Ouais. Ah, ça fait depuis là-bas. J'arrive pas à voir le lien.
C'est lequel du message du d'accord?
Sur la diversité dans la roue.
Ah non, je n'ai pas vu.
Ah non, j'ai pas vu. Voilà.
Vas-y, pour voir le lever. Pour sa réponse.
Ah, j'ai le lien.
Tac.
No problem. Waiting.
Et ben, on va pouvoir commencer ce séminaire. Donc, bonjour à tous. Merci de nous avoir rejoints pour ce nouveau séminaire. Donc, on a la chance d'accueillir un invité, donc Jácint Tökölyi. Et tout d'abord, donc c'est Massiva qui va vous présenter la minute verte qui concerne la climatisation. C'est là.
Merci Sophie. Bonjour à toutes et à tous. Pour rester un peu dans une thématique très d'actualité à partir de ces débuts de l'été, et ben du coup c'est la climatisation au travail. Donc là je l'ai intitulé au bureau, mais c'est des gestes qui sont tout à fait pertinents dans les laboratoires également. Donc d'abord quelques chiffres, savoir que selon l'ADEME, la climatisation représente 20%, consomme 20% d'énergie, et au niveau de la DRO de l'Occitanie l'an dernier, cela représentait 9% de la consommation. La climatisation contribue au réchauffement, au réchauffement des villes en augmentant la température entre 0,5 et 2 degrés, puisque ça rejette de l'air chaud, comme vous le savez. Donc, comment préserver la fraîcheur dans ces lieux de travail? Donc, d'abord, le matin en arrivant, dans la mesure où il fait frais, on aère. Et puis, derrière, on va fermer dès qu'il commence à faire chaud, en utilisant des protections solaires adaptées, dont normalement les bureaux sont équipés, ainsi que les locaux, les labos. On peut réduire également la chaleur en utilisant moins d'appareils électriques en permanence, et notamment certains éclairages d'appoint. Et enfin, penser à utiliser les ventilateurs à la place de la climatisation dans un premier temps. Ça peut aider grandement. Donc, par contre, si on ne peut pas se passer de devoir mettre en place la climatisation, à ce moment-là, il ne faut pas l'allumer et laisser la fenêtre ouverte. Pensez à la régler vraiment à température pas plus fraîche que 26 degrés. Et puis, minimiser les écarts avec l'extérieur, puisque sinon, on a des chocs thermiques. Et pour finir, quand on a fini sa journée, il faut penser à l'éteindre. Voilà, et à chaque fois qu'on quitte son bureau et les labos à la fin de la journée. Voilà, je vous remercie pour votre attention et bon séminaire.
Super, merci beaucoup.
Je te laisse juste, j'ai peut-être la clé.
Je vais gérer. So now we'll start the presentation for today. We have the pleasure to have Jácint Tökölyi, who will present us a presentation on sex and death in Hydra. And I let you introduce yourself.
Okay, thank you. Can you please help me to get the screen?
Wait.
Great, thank you. So thanks very much for having me here. And I think instead of presenting me, I will start with presenting the Hydra because those are much more interesting. And no, I can't. Okay, I found it out. Thank you. So actually, I share a passion with these guys here with Hydra. So I will start with saying that Hydra are really, really cool creatures. And it's actually a very small genus of freshwater cnidarians that live across Europe in most types of water bodies. They have a really simple body organization. So the animal is just like a tube with some tentacles at the end, and it has two cell layers, an ectoderm and an endoderm. But when you start to look into these two cell layers, this is where the interesting details start to emerge, because most of the body is composed of stem cells that are capable of self-renewing and to differentiate into novel cell types. So the Hydra has three families of stem cells, the ectodermal and the endodermal stem cells, which are able to divide and continuously renew themselves, and the interstitial stem cells, which are smaller, and they are also renew themselves, and they are also responsible for giving rise to other cell types within the Hydra body, such as the nematocytes that are involved in food capture, the nerve cells, or the reproductive cells, the germ cells. So it's actually a really small number of cell types are present within the organism. So they are relatively easy to quantify, to study under a simple microscope. And due to the fact that they have such a huge proportion of stem cells inside the body, Hydra have a remarkable capacity of tissue plasticity. That means that you can cut a Hydra into two pieces, like here, and they will regrow the lost pieces. And you can also take them apart into cells and then just let them to grow together. So they just found the cells each other and can reaggregate to form a new individual. So it's a really huge tissue plasticity that's present within this system. And I'm more an ecologist, so I'm interested in diversity in the natural world, and why do species or individuals in a population or species in a given habitat differ from each other. And Hydra is really good for that because they have a really diverse life histories. For instance, if we look at aging, we see a huge difference between aging patterns within species and between species. Actually, Hydra became quite famous when a few years ago research groups in Germany and the US published a paper which was nothing just counting hydras to 7 years and keeping them alive and looking at their survival and reproduction patterns. And this is the figure that they got after 7 years of meticulously maintaining Hydra in the lab and recording any occasion when someone died or produced an asexual offspring. What they found that basically within 7 years, these creatures don't really show an increased rate of mortality with time. So normally what happens with an insect or a vertebrate is that as time progresses, you get an irreversible increase in the rate of mortality or reproduction, decrease in reproduction. So that's aging, basically. That's the definition of aging. This is something that doesn't seem to happen in this species of Hydra vulgaris. They can be maintained in the lab for a really long time. And from this survivorship trajectories, the authors estimated that the expected lifetime of these animals in the lab under constant and perfect conditions could be from 1 to 10,000 years. It's really impressive. And there are some species like this, and they are called collectively a species with negligible senescence. So it's present, but it's present in very low rates. But then there's another species of Hydra, Hydra oligactis, which has been studied parallelly, and this has been also maintained in the lab. And researchers in Japan have studied them under not under asexual reproduction, but sexual reproduction. So this is a species where you can induce sexual reproduction by putting them into cold temperature, cooling the environment. They start to undergo sex, and this is the trajectory of mortality that you can see. It's basically at the end, after several months, it's 100% mortality within that batch of laboratory individuals. So in this case, there is a very fast increase in mortality, and this is what we call catastrophic senescence. This is also present in salmon, for instance, and in some other vertebrates like marsupial mice, males also have this kinds of accelerated senescence. So this is really interesting. Within a same system, we have a stage or a category where they reproduce asexually and have no senescence. And there is a stage where they reproduce sexually and they have accelerated senescence. And this can actually be combined within the same life cycle. And here I'm focusing on Hydra oligactis. We are working with Hydra oligactis because we don't have time to wait for vulgaris to show senescence because it's 1,000 years. So we thought that maybe we focus on oligactis because it's a bit shorter. So all the work that I present in continuation will be about Hydra oligactis. And so just a short introduction about the species. This is a temperate species, so it lives in Europe and North America and Asia and northern latitudes. Here around Montpellier, there are several populations. So this is about the southern limit of the distribution. So they live in highly seasonal environments. There is a relatively warmer summer where they reproduce asexually. And as far as I know, we know there is little senescence during this time, at least much less during the asexual sexual phase. And they start to switch to sexual reproduction during the autumn when the temperature goes down, and they produce resting eggs. It's just like in many other zooplankton like Daphnia. They sense that the cold is coming and there is a risk of freezing, and they start to produce resting eggs that can survive the winter. And that's when sexual reproduction occurs. And this is what's followed in the increased rate of mortality in this system. So our questions are really simple, and I would really like to understand why is that we have a species that is actually capable of biological immortality, but under some conditions loses that ability and undergoes a very quick senescence. And we're interested in the ecological aspects and the evolutionary aspects of that. And also we'd like to understand the functional mechanisms behind that, the gene regulation, physiological regulation, but I'm going to talk less about that today. So one of the earliest things that we did when we started was that we wanted to find out whether this kind of increased or reduced somatic maintenance and increased senescence could be observed under natural conditions. Everything so far has been done in the lab. So we wanted to see if we see a similar pattern under natural conditions as well. So we went out to the field to a nice site with lots of Hydra, and we did a time series collection from early autumn to late autumn or early winter, where we just went out, collected Hydra individuals and looked whether they are sexual or asexual. And what we found is that there is a polymorphism in the population. So you can see in early autumn, you can see sexual individuals, and by early winter, the proportion of sexual individuals decreases. But it's a polymorphism. So you have both sexual and asexual individuals within the population throughout the sexual period. And we brought some of these individuals to the lab, and we did experiments with them, really short-term experiments. So everything lasted for a week. So they had the lab, they didn't have the lab conditions. They experienced the field conditions, but we did regeneration experiments in them. This is really simple. You just use a scalpel to cut the head of the Hydra, and normally it can regrow it. But what we observed is that if we collect sexual and asexual individuals, then this is a sexual male, and this is a sexual female. You can see the X here, and this is an asexual or not reproducing individual. You can see that after a week, the non-reproductive or asexual individuals are able to regrow their head, but in the sexual individuals, there's no regeneration. It's like switching off regeneration entirely. And this is supported by the proportions. You can see that in adult males and females, there's basically no regeneration at all. And if you look at the cellular composition, it's quite easy to take these animals apart and count different types of cells. We counted nematocysts, so stinging cells, which are important for food capture, and interstitial stem cells, which are the somatic stem cells of the Hydra, with the idea that we expect a lower rate of cell types involved in somatic maintenance for the sexual individuals. This is what we found. So this is the number of nematocytes, nematoblasts that were present in sexual individuals, substantially lower than in the asexual individuals. So, and the same with the interstitial stem cells. So the conclusion here is that sexual individuals have reduced investment into somatic cell types and reduced regeneration capacity, which could explain their reduced survival rate. We couldn't, of course, be able to really follow survival in the field, but this shows at least partly that the difference between sexual and individuals persists under natural conditions. So what we wanted to find out next is that it's really interesting to see the sexual and asexual individuals in the field together with each other. Why is that? So why is within the same population, under identical microclimatic conditions, you see a sexual and asexual individuals, although these two phenotypes have an entirely different trajectory. So one will die quickly, the other one will survive relatively easily. So we wanted to see whether there's a genetic structure underlying that pattern, and we tried lots of population genetic methods, but what worked at the end was a method called restriction site associated DNA sequencing, RAD-seq for short, which is just you take the genome of an individual and you subsample a small portion of it, like 1 to 10% of the genome, and you sequence that with NGS, and that's enough to genotype individuals. And we collected lots of Hydra from different populations in Hungary. You can see with red colors, it's sexual individuals, with blue or white, that's asexual or non-reproductive individuals. So it's quite variable. Somewhere we have more sexual individuals, other times we have less. And then we had both sexual individuals and asexual, and we genotyped them. This is what we got first. This is a complicated graph, but it basically shows the relationship between Hydra polyps collected on the field. And each of these rows and or batch of rows is a population. So here are our populations, and the relationship within a population can be seen as this triangle, and the relationship between populations can be seen in these rectangles. And the color shows the relatedness of the polyps. And we were not really surprised to see that within a population, we see very high relatedness. Sometimes we detect clones within a population, but we were surprised to see that overall, there is a very small genetic variation within the population, and there is quite a strong relatedness between populations. So within Hungary, a few 100 kilometers are basically not too much. 200 kilometers, you can still have second order relatives between the populations, like cousins. That was really surprising to us. But more interestingly, when we combine this genetic structure with information of the sexual state, reproductive mode of the individuals, we find that there are sets of clones. So these are sets of clones because they are genetically very similar to each other. They cluster on a single branch. And with different colors, you can see the sexual and asexual individuals. And then there is no genetic structure with respect to reproductive mode. You can see sets of clones, so polyps that are genetically identical or almost identical. And one of them is sexual, the other is asexual. So the same genotype is able to produce in the same population at the same time, both sexual and asexual phenotype. So we wanted to find out what are the factors that generate that. So there seems to be relatively little genetic background to these differences, but there seems to be a lot of phenotypic plasticity. So the same genotype capable of producing different stages depending on something that we didn't know at the time. So we started out with different factors. One of the factors was to look at the age of the polyps. So age is a bit difficult to define in a Hydra because you can measure age from hatching from the egg, but you can also measure age from an asexual polyp after it detaches from the parent. So we focused on this asexual age. So we took polyps with a bud, and then we waited until the bud detaches, and then we took the freshly detached bud and the parent, and we put them into the cold temperature to simulate the winter, and then see whether we find a difference. And the difference we found, here are the males, and here are the females. And the top row is an adult individual, adult male, and a juvenile or a bud male, an adult female, and a bud female. And with the adults, you see the typical pattern. So you cool them, they start to develop testes like this. And then after developing testes, after a few weeks, the photos goes for 10 weeks, they start to degenerate. They decrease in size, they lose their tentacles, become smaller and smaller, and eventually some of them become to zero size. Not the same in the buds. So the buds first seem to grow a little, and then have a few reproductive organs. You can see that much fewer here than here. And then they don't decrease in size, and they just regenerate after sexual reproduction and follow asexually after that. So sort of the same pattern is seen in the females. And we also try to collect data on more detailed level. So we looked at the reproductive rate, number of reproductive organs in males and females. And we also looked at the cellular composition, the number of reproductive cells and its dynamics through the sexual period. And we also looked at regeneration and stem cell numbers through the sexual reproduction period and survival. And in every respect, we find the same pattern. So for sure, the juveniles, the buds, have a lower rate of reproductive investment, fewer gonads. They also have a delayed sexual maturation. So this is adult compared to juvenile. You see a delayed maturation of, these are basically sperm cells, the same in females. And they have a difference in regeneration capacity and a difference in the number of stem cells. So juveniles have a lower reproductive investment and a higher survival rate and a higher maintenance of somatic tissues. So this could be one of the explanations that explains why we see a difference between within the same genotype. And we saw that there's a difference in the body size of the two types of individuals, buds and adults. So we wanted to find out whether this is something more important. So is there a body size effect in reproduction and survival? What we did is to go out to the field and collect strains from the sample from the natural populations. And I'm really proud of this experiment. We did this for 2 years. We didn't do anything else during this period. We had 181 strains established with a total of more than 1,000 polyps. And we followed each of them for 5 months individually to record reproduction and survival rate, and also body size and age. And what we found is that there is a correlation with body size after controlling for age. So larger males and females have a faster sexual development. So they need less time to develop gonads. They have a higher fecundity, so more testes and more eggs. And they also have a lower survival rate, both males and females. But we didn't stop here, because with Hydra you can do really nice or nasty, depending on your how you see it, experiments. We wanted to see whether the body size effect is causal or just correlative. So we did an experiment where we cut out pieces from one Hydra, like here, and pieces from another Hydra that differed in size, and we switched them. We grafted them together. They heal back. They actually like this procedure. It's no problem. And these are clones. So you can switch tissue between members of the same clonal lineage. And as a result, we had this enlarged and reduced individuals. And then we just put them to the code to see whether there is a difference in sexual reproduction and survival rate between them. And here is what you can see in this experiment. How do I go back? Like this, okay. So these are the small individuals and the large individuals at the time of cooling. After 3 weeks, you can see a difference in sexual development between the reduced and enlarged polyps. So the reduced ones take more time to maturate the gonads. And they also have a difference in fecundity. So the enlarged individuals have larger fecundity, both in males and in females. There seems to be some sex differences, but we don't know if these are consistent, because we work with only one male and one female strain. And we see a difference in survival. So polyps with reduced body size have a higher survival rate. And we can actually experimentally prove that it's body size itself that's causing the difference in survival rate, and it's not something correlated with body size. And so we know that age and size affects plasticity in reproductive and survival strategies in Hydra. Next, we wanted to see if there is a consistent pattern in that. This was really annoying when I started working with Hydra, because quite often we went out to the field, collected individuals, made an experiment, and then we went out back, did the same, and got different results. And yeah, so how do you interpret that? The same population, sometimes you have different results. And what I've learned from this is that quite often, if you are not able to repeat experiments, there is a biological explanation for that. And I think we were able to find the biological explanation for these differences by basically repeatedly sampling the population, a single population, which we sampled during the spring and during the autumn 2 times, so 2 years, spring and autumn sampling, establishing strains from them and comparing the proportion of sexual and asexual individuals within these strains. And what we find is that in the spring collected samples, we see a relatively high proportion of asexual reproduction. And in the autumn collections, we see a low proportion of sexual reproduction. And there is also a difference in the speed of sexual development between the 2 groups. So in the autumn, they are really fast. In the spring, they are really slow. And I think this makes sense. So if you're a hydra in a lake in the spring, there is a very low chance that the next day there will be a really cold spell and the winter will start, like in March. No chance that there will be a winter tomorrow. While in September, there is quite a large chance that the winter will start earlier, and then you have to produce the resting eggs, otherwise you won't be able to produce them. So it's something like a preparedness that is present at the level of the hydra polyp within its germ cells, probably, that enables them to prepare, and that increase that rate of preparation as the season progresses. What could be the factor behind this increased preparedness? We, of course, suspected its temperature, because the summer is hot, so you expect that warm temperatures could increase the rate of sexuality in these creatures. So we did a lab experiment. This involved 2 cycles of cold and warm treatment, so it's a bit more complicated. But basically, we took these hydra polyps, induced sex in them, and then after that, some of them survived. You could see that survival rate is sometimes occurs, so there's sometimes survival. So we can take these survived polyps to generate a strain that is propagated asexually in the cold. So it's like the winter for the hydra. They are asexually propagated in the cold, and then you just expose them to warm temperature and cool them again to see whether they are able for sexual reproduction or not. And what we saw is that if they are kept continuously in the cold, they are not capable of sexual reproduction. If they are exposed to a short 1 week of warm exposure, then there is some increase in the rate of sexuality. And if you expose them to 4 weeks of warm treatment, then you see a large increase in sexual reproduction. You can see this with gray. So probably warm temperature has a causal effect in determining the ability of these hydra polyps to reproduce sexually. We are also continuously interested in the microbiome of hydra, because they seem to have a really tight relationship with microbes, and we wanted to see whether microbes the microbes that live together with hydra could be responsible for generating these differences in sexual and asexual phenotype. And we had again a relatively large sample of several hundred hydra polyps, for which we had information on the sexual phenotype and also microbiome composition from field collected individuals. And we compared them, but didn't really find a difference. So it seems that, at least under the conditions that we collected them, there doesn't seem to be an effect of the microbiome on the sexual phenotype of hydra, which we found surprising, because there's indeed a really complex interaction between hydra and the microbes. For instance, the sexually developing females emit lots of antimicrobial peptides that determine what kind of microbes can go into the resting eggs. Still, we weren't able to show there is a difference, but it could be due to the fact that it was just in early periods of the sexual period, so maybe we didn't do it at the right time. We plan to repeat this. And finally, a bit more speculative words about the reason why I'm here is to think about tumors in Hydra. And Hydra are able to, or have been shown to develop tumors like these outgrowths that you can see in these polyps, which have been shown a few years ago. And it seems to be a connection with the sexual phenotype, because the cells within these tumors are actually germ cells. So the germline stem cells that are responsible for producing the eggs, under some conditions, produce tumors, which can affect hugely these animals. So we're not really sure what these tumors are, but what we know is that at least in one of the strains, this is a microbiome-mediated overgrowth of sexual development. So two species of microbes that are present in the Hydra release some kind of chemicals that simulate or make the Hydra germline stem cells to proliferate. And this will result in this tumor development. And yet we are just trying to find out what could be the reason for that and how it is connected to the overall Hydra biology. I really liked what Fred said multiple times about host parasite, host tumor interactions, that you can divide it into basically three ways. One is that, so to explain a phenotype that is the result of a host parasite or a host tumor interaction, you could explain it like some kind of adaptive mechanism from the part of the host to reduce the effects of the infection and increase its own survival rate. You can also explain it with some kinds of parasite manipulation to increase the own transmission rate. And it could be also something that is not related to either. I'm not sure which one is currently the most likely explanation, but we have to find it out. So I'm happy to be here and discuss about that. So just as a summary of what we have found out about Hydra, it's Hydra oligactis. It's much more complicated than was originally in these two papers. It's not like you have an asexual stage and a sexual stage. Within the asexual stage, you have an asexual stage which has a low capacity for sexual development, and a stage that has a high capacity for sexual development. The first during the winter, the second during the summer. And then you have a sexual stage. This is where tumors appear, as far as I know. And it's much more complicated than like you have sex, you die. It's not like that. Not in Hydra either. So it's more complicated. You have a range of phenotypes. You can have a highly sexual phenotype that has a high rate of mortality, and the phenotype at the opposite hand with a low rate of sexual investment and low rate of or high rate of survival. And it seems to be not entirely genetically determined, at least these differences, because there is a huge phenotypic plasticity in that. And then the age of the polyps, the size of the polyps are the factors, and warm exposure are the factors that mediate these differences, at least as far as I know, or we know. And with that, I would like to thank you again for having me here, and just briefly mention a large number of an army of really fantastic students that actually do my work while I'm here, working in this fantastic city. So thanks very much.
Question time. If anyone
Yes, please.
Many thanks for your presentation.
Thanks for coming.
Really cool and fascinating system, really. Thank you so much. So I have a couple of questions. So I think you said that at the end. It's not because you had sex that you will die. And during your talk, I was wondering whether all your results can be recapitulated by just saying that you just have a trade-off between sexual reproduction and survival. But you said no.
Yes.
I'd like to come back on this point for question 1 that you asked. You said that sexual individuals have reduced somatic functions on question 1. And I was wondering if we can come back to question 1. Here, so on the picture, so the C is the male, right?
Yes.
And I was wondering whether you know here whether the males have already reproduced, or are they going to reproduce? Are they already had an episode of sexual reproduction here? Or?
Well, these ones probably have already released sperms, and they continuously do that. So in early stages of sexual reproduction, their regeneration rate is not affected as strongly. There is a stage here which we call sex undetermined. That's because these are the immatures which look alike. So these are males or females and in early stages of sexual development, and they have a high rate of regeneration. And so the peak reduction in regeneration is when they have most gametes production.
So it looks like they die when they are already reproduced, right?
Yes.
I have a related question about question 3, actually. Here, I was wondering, so it's about the age, I think.
Yes.
If you take some polyps, just the previous slide here. So you have your 2 groups of polyps.
Yes.
And so the big one, are they already reproduced here?
Sexually, no.
Oh, that's interesting.
Sexually, no. So the difference starts to emerge, for instance, let's see, in some kind of somatic maintenance. This is the number of interstitial cells in males and females. So in the juveniles or the buds, the number of interstitial cells is actually starts to increase first, while in the adults, it decreases very quickly. And basically, we think that this is because, I'm coming back to first slide, this trade-off is caused by the fact that the cell types that are needed for reproduction and the cell types that are needed for self-maintenance, so the nematocytes and the nerve cells and the gland cells, derive from a single cell population. So you can either have reproduction or you can eat, but you cannot really do both at the same time, especially if you are constrained in time because of the cooling and the coming of the bad weather. So this is what we think an explanation for the trade-off.
How about the distinction between the relative contribution of age and size in your experiments? So the older Hydra are also bigger?
Yes.
Right?
Yes.
But I guess at some point they reach the same size but can have different age. And that would be cool to repeat the experiment with different age but same size.
Very good idea. Yes, we haven't done that yet. I think both age and size could contribute, because at least in the correlative studies, we control for age, and then we still see a size effect. And the size is there for sure because we have manipulated it. I think it would be possible to manipulate the size of younger polyps and to get a polyp that is the same size as a big one, and then see the results.
My next question: can we reconstruct individuals like that of different growths? Or they must be the same?
No, it's possible, and actually within species they don't seem to have any problem with incorporating tissue, genetically distinct tissue from the same species. They also incorporate tissue from a foreign species, but they start to phagocytize each other. We haven't tried that, but it's a really interesting idea. Yeah.
My last question, just out of curiosity, what's the sex determination system?
As far as we know, it's determined by the type of germline stem cells, so the gamete precursors that the polyps have. So it's not present in this graph, but between the eye cells, the interstitial stem cells, and the germ cells, there is a germline restricted stem cell population, and the polyps carry that population of stem cells with them when they reproduce asexually, and that maintains a stable sex in the polyp. But if they lose it in some way, for instance, we can expose them to heat stress and then they die, and as a result they can regenerate the germline from the interstitial stem cells, and then we will get sex changed individuals sometimes.
Male or female cannot do both.
Normally, yes. Unfortunately, some of our animals haven't read the scientific literature, so they sometimes are hermaphrodites, but normally it shouldn't be present because there is some sort of masculinization. So if there are male germline stem cells in the body, then they suppress the female germline stem cells. And so if you remove them, you get a female. Thank you.
Really interesting the results about the size effects, especially because we look at this with tumorous individuals, and we saw that even smaller individuals without having tumors have a higher sexual reproduction than the ones who have tumors, so they are bigger. So it's very interesting because our control behave like in your study. They need times to grow, to be big, and to make a lot of gametes. But the tumorous ones behave at the opposite. When they are younger and then smaller than when they have these big tumors, they are able to produce more gametes in a smaller time.
Maybe it's because just an idea, because the large tumorous individuals are maybe in a later stage of tumor development, and then maybe that impacts negatively impacts the polyp, so they will be more stressed if you cool them. Just an idea, but this is really interesting observation.
Because maybe it was used because of the investigation of the strain that without any cooling.
Yeah, or we could do size manipulation on the tumorous individuals.
Yes,
Yeah.
But also because we made graft of other lineages. I showed you the rhesus, but we also did the asexual budding, and we saw that even just the budding rate can be really impacted by a small graft of another lineage. So maybe there is also a mechanism that can trigger more reproduction.
So based on this, Justin told us yesterday that if she introduces tissue from a different species, right, a different lineage into a Hydra, then the asexual rate of reproduction increases. So maybe they are somehow able to sense that there is a foreign tissue and then just try to divide. Or
but it depends on the donor. Some lineages decrease, some decrease.
Use a single strain.
Yeah. Maybe it's because also the robusta are bigger than the oligactis.
Could be.
concerning the few individuals that do not immediately die after the sexual revolution, how long do they live after?
So if they regenerate and we keep them in the cold, I think we never tried, but we never see them dying. So I think it's the same pattern of lack of senescence that is present in vulgaris. So I would say there's no senescence there.
And do you think that if you have a second episode of sexual reproduction this time?
Yeah, I would say yeah. So it's totally linked to sexual reproduction. Okay.
Do you have the thing that beyond age and size, Hydra could have a memory or a time memory of the season? Because we have the thing, it's like a tune that we observe more spontaneous tumor in autumn or winter. And we work on Hydra, so they never see natural field from a long time ago. And we were thinking that maybe they still have some kind of memory of timing. And they produce more tumors, spontaneous tumor, at this time when normally sexual reproduction occurs. And your graph with the delay of production of sexual organs, what do you think about it?
Temperature definitely is a factor. So more exposure to high temperature affects that. But I think I have bad news for us in other ways as well, because there's been a study where someone hedged the Hydra from the egg and then kept them for 3 years under constant conditions. And it saw a constant increase in the rate of sexual reproduction without no exposure to environmental factors. So there might be some other factor that increases the rate of sexual preparedness. And they are really sensible to things that we don't, we are not able. There's another study where someone kept them under constant conditions, and they saw the repetition of seasonality in the lab. So it turned out that they used tap water, and the Hydra was able to sense the carbon dioxide concentration in the tap water to just follow the normal seasonal dynamics. They have different view of the world than as us.
Perhaps the night and day length can.
But yeah, we didn't try that. So actually that could be a good idea, but in this experiment, it was constant. Yes. For us, when we do the sexual induction, we switch from a 12-12 to a 8-16. So it could be cooling or photoperiod. So we actually use a double treatment. But I think it's more the temperature that affects them, but we haven't tested that. Okay, thank you very much for being here.
Eh ben, merci à tous d'être venus. Et donc, on va terminer ce séminaire et on se retrouve donc avec plaisir la semaine prochaine pour un ou deux nouveaux séminaires. Merci à tous.